In this slide, we're going to pick up where we left off at the end of the last video, talking about the initiation phase of transcription. In the last video, we talked about this particular length of DNA that represents a gene. It has a protein coding region that will be transcribed. It has a regulatory region that includes the promoter. And we talked about how the RNA polymerase enzyme will bind to the promoter so that it can begin transcription right at the beginning of the protein coding region. Now let's look at the bottom part of this drawing. If you look very carefully, I know it's faint, but if you look very carefully, there's sort of this very light gray outline of a structure right here. And there's a second outline here indicating some of the shape, some of the topology of this big molecule. This light gray area represents the RNA polymerase enzyme. And you'll see that it has come in and it has landed on the promoter in the regulatory region of this gene. It has split the double-stranded DNA molecule so that the strands are separated from each other. We're now at the end of the initiation phase, and we're ready to begin the second phase, which is elongation. On this slide, up at the top, we can see that double-stranded molecule of DNA that has that yellow color, telling us that it's the region of a gene that codes for a protein. We can see that that light gray outline of the RNA polymerase enzyme has moved forward. It is now about halfway down the length of this gene, and it is creating an RNA molecule. Specifically, in this case, a messenger RNA molecule that is shown in red. Now, it's hard to tell from the diagram, but this enzyme started over here. It opened up the double-stranded DNA and it started to move this way, in this direction, down the length of the protein coding portion of the gene. It's now about midway, and it will continue to move forward down the length of the gene. We give this a name. We call this direction downstream. We say that this enzyme is moving downstream from what we would call the very start of the protein coding region to its end. Now notice that what's behind the enzyme now, what is now upstream from the enzyme, is back into its double-stranded shape. In other words, the enzyme must have opened it up, transcribed the DNA, making some RNA from it, again shown in red. And then as the enzyme continued forward, what it was finished with returned to its double-stranded structure. That will continue as the enzyme moves forward downstream again. What it has finished transcribing will return back into its double-stranded structure. The RNA that's been made will sort of hang off the back of the enzyme, as shown in the image. Now, there's something else important to notice in this picture. The RNA is being made on one of the two strands of DNA, just one. Only one of the strands of DNA has the code in it to build a protein. We refer to that strand as the template strand. This is different from what we saw in DNA replication. In DNA replication, the entire DNA molecule has to be replicated, has to be copied. So both strands of DNA have to be replicated. During transcription, only one DNA strand is used, the template strand, and only the area of one gene that codes for one protein is being transcribed. We're building up a protein during gene expression that the cell has a need for at this particular moment in time. The template strand, again, has bases in it that are in a particular order or a particular sequence to build a protein. The RNA is going to be a transcript of that DNA. It's not going to be an exact copy. It's going to be a complementary copy. Where there's an A in the DNA, there will be a U in the RNA. Where there's a T in the DNA, there'll be an A in the RNA. Where there's a G or a C in the DNA, we'll find the complementary C or G in the RNA. It's a transcript. Now, the other strand of DNA, the one opposite the template strand, also gets a name. It's called the coding strand. And this can be confusing for students because it sounds like the code to build the protein ought to be in the coding strand. But that's not the case. The RNA that's made during transcription is made from the template strand of DNA. And as the enzyme starts traveling down and using this template strand to build RNA, this is the part of transcription that we call elongation. Now take a look at the information in the box at the bottom of the slide. All they've done here is they've taken this region of the DNA and they've added in actual bases instead of just these little sawtooth lines that they've drawn. So you can see we've got a coding strand at the top and you can see the bases. We've got a template strand at the bottom and you can see the bases. And we're building RNA on the template strand. And they're showing us this complementary nature. They're showing us that if there's a T in the template strand, the RNA polymerase enzyme will place an A in the RNA. If there's an A in the template strand, the RNA polymerase enzyme will place a U. Remember, there's no T in RNA. Now, the last part of transcription is called the termination phase. And as we said in the last lecture, during the termination phase, the RNA polymerase enzyme is going to come to the end of the protein coding region of the gene, and it's going to hit another regulatory area. It's called the termination signal. All it is a piece of DNA. It doesn't look any different than any other DNA. It's just that the bases that are found in the termination signal are going to cause the RNA to do something different. So the polymerase enzyme will have continued down through the yellow protein coding area, building RNA from the template strand, and then it will just continue on into the termination signal, building RNA on the template strand. But here's the difference. The bases in the DNA that are found in this termination signal, all the way along this blue area, the bases in the DNA are in such a particular order that the RNA that gets made from it will fold back on itself in this region. And that's what they're showing us in the picture. The enzyme has traveled along into this blue termination signal in the DNA. It's still making RNA. It's still doing its job. It's building RNA off of the template strand. It's just that the way the bases are ordered will allow the RNA to loop around and bind to itself in this area. It's just due to the order of the bases. In other words, there has to be some complementary bases that can line up and bind to themselves, essentially, in that region. Now, this is what's going to stop the elongation phase. This is what will stop transcription, because this big looped piece of RNA is going to gum up the works, if you will. It's not going to be able to just hang off the enzyme like the rest of the RNA has. It's too big. It's too bulky. What's going to happen is this loop that forms spontaneously, this loop is going to cause the RNA polymerase enzyme to just let go at this point, and transcription will end. We've looked at this image before. This just shows us in a drawing the process of gene expression, the central dogma, really. Remember, we start with DNA, and that DNA, the double-stranded molecule, is going to be used to build RNA, the red. That's a process called transcription. The RNA will then be used to build a polypeptide. Remember, that's an unfolded protein. That polypeptide will eventually fold into a protein. So RNA is used to build, eventually, a protein. That's the process that we call translation. And it's that protein that ultimately forms that will give the organism a trait. So transcription has 3 phases. It has an initiation phase, where the RNA polymerase enzyme locates and binds to the promoter region in the regulatory portion of a gene. Then it has an elongation phase, where the RNA polymerase enzyme will travel along the length of the gene, the protein coding region of the gene, and it will build an RNA off of the template strand of DNA. Then we go into the termination phase, when the RNA polymerase enzyme starts to transcribe the termination signal in the DNA and creates a portion of the RNA that will fold back on itself. That creates a loop of RNA that no longer fits in the RNA polymerase enzyme, and the whole process stops. There are a couple of other things that we should know about transcription specifically related to our microbes. In prokaryotic cells, in bacteria and archaea, transcription is occurring right in the cytoplasm. And that's because the DNA, as it sits in the nucleoid, is right in the cytoplasm in a prokaryotic cell. In eukaryotes, it's different. Eukaryotic cells have organelles. So in eukaryotic cells, the DNA is housed in the nucleus, and transcription will occur in the nucleus in those cells. But in prokaryotes, it's all out in the cytoplasm. So what happens in a prokaryotic cell is that while transcription is happening, as soon as a short length of RNA has been made, translation will go ahead and start. As soon as there's a little bit of messenger RNA produced, translation will begin. Ribosomes are going to attach to that growing piece of messenger RNA, and they're going to start this process called translation. When you look into a prokaryotic cell during gene expression underneath an electron microscope, you can actually see the fact that transcription and translation happen simultaneously. And the reason you can see it is because of the ribosomes. As soon as a little portion of mRNA is produced, ribosomes are just going to start translating it. And remember, the cell is not going to make one copy of the protein from this mRNA. It's going to make as many copies of this protein as it can. It will, in other words, translate this messenger RNA as many times as it can, building as many molecules of this particular protein as it can, until that messenger RNA degrades, goes away. So that means that it's not one ribosome that's going to jump onto the mRNA and translate it. Many ribosomes are going to jump on, one after the other, each ribosome reading the transcript, reading the messenger RNA, and building a protein according to the code that's in that messenger RNA. And under the electron microscope, you can see this. We refer to these as beads on a string. Each ribosome looks like an individual bead jumping onto this string of messenger RNA, creating what we technically call a polyribosome. On this slide, down at the bottom, you can see a drawing of this process going on. And up at the top, you can see an actual electron microscope image. You can actually see these beads on a string. Let's look at the drawing first. The blue color represents the double-stranded molecule of DNA. We can't see the RNA polymerase enzyme here. Because of the scope of our view, we're not able to see down at the level of the enzyme. But we know that somewhere in this blue line, there's an RNA polymerase enzyme, and it is traveling along the length of the DNA, separating the two strands, making RNA off of the template strand. There's not one RNA polymerase enzyme doing this. There are several copies of RNA polymerase enzyme doing this, one after another after another. So there's an RNA polymerase enzyme right around here, sitting in this DNA. I know that because I can see the red messenger RNA coming out of it as it's being produced. Over here, there has to be another RNA polymerase enzyme because here's some more messenger RNA being produced. You'll notice that this piece is shorter than this piece. That tells me that the direction of transcription, it must be going this way. In other words, the start of the protein coding region must be here, and the end of the protein coding region must be here, because the RNA, the red line, is getting longer and longer. So I've got 1, 2, 3, 4 molecules of RNA polymerase enzyme working on this gene right now, transcribing it. And all 4 of them will eventually travel down and create a long messenger RNA, again shown in red. Notice that there are these little circular beige structures jumping on that red line. These are ribosomes, individual ribosomes. They've taken this one for us and they've blown it up. So here's the ribosome sitting on the red messenger RNA molecule. And there's a little white string of beads, that's what it looks like, coming out of the ribosome. This is the polypeptide being built. Remember, just like the RNA polymerase enzyme is traveling along, sort of decoding the DNA, the ribosome is traveling along the messenger RNA, decoding it and building a polypeptide out of amino acids. There's not one ribosome per RNA, there are many of them. We're going to get many copies of protein made from this gene. All right, a cell, when it needs a protein made, it doesn't need one copy of that protein made. It doesn't need one molecule of that protein made. It needs many molecules of it made so that it can be used in the cell. So every time gene expression happens, every time a protein is made from a gene, we're going to get multiple copies of that protein made.